what we do know is that in normal development, there is synaptic pruning in order to get rid of less active synapses and preserve more active synapses to produce more efficient brain action and the appropriate circuit development. And in neurodegeneration, we have this dysregulated C1q deposition, and as a result, neuroinflammation develops and cell loss occurs. Now, there seem to be various triggers of this dysregulated C1q deposition. For example, in Alzheimer's disease, amyloid seems to be one of the triggers. Cell debris can be a trigger. In Huntington's disease, we really don't know what the trigger is. We know that it's present if one looks at post-mortem studies of pre-manifest or manifest individuals with Huntington's disease. You can see that this is present. You can also see this is present if one develops a transgenic mouse model of Huntington's disease. And we know that we can block this and preserve function if you either knock out C1q or you administer to the mouse the C1q antibody. So how it actually occurs and what's the trigger in Huntington, we don't know. We know it's present, and it seems like preventing it, at least in our preliminary human study and in the animal models seems to make sense and be effective. It's very different, of course, than the primary genetic therapies, such as ASO therapy, small interfering RNA studies, or CRISPR studies. Very different in that we are not going after, if you like, the primary genetic defect. So this is probably a downstream phenomena, and it seems to be a common phenomena in multiple neurodegenerative diseases. So this, we anticipate, can be applied in other neurodegenerative diseases, and indeed, phase 2 studies in ALS are ongoing, and there are studies of this antibody in other diseases, such as truly peripheral inflammatory diseases, such as Guillain-Barre syndrome. A phase 2 study has been completed. A phase 3 study it will be beginning shortly. And in non-neurological diseases, such as cold agglutinin disease, the preliminary results are really quite excellent.